Welcome back to the deep dive. Today we're shifting gears a bit. We aren't talking about tech or history or economics. We are talking about a kingdom.
A very misunderstood kingdom.
Right. And I don't mean a political one, I mean a biological one. It's a kingdom that is fundamentally responsible for life as we know it on Earth, yet simultaneously represents this growing, shifting, and frankly terrifying nightmare for modern medicine. We are talking about fungi.
It is a massive topic. And honestly, it's one of the most frequently misunderstood areas in all of microbiology. I mean, we tend to treat fungi like they're just, I don't know, weird plants or maybe complicated bacteria.
Yeah, just germs.
Exactly. But biologically, they are vastly different from both.
Yeah.
They occupy their very own distinct, incredibly complex space in the tree of life.
And before you check out thinking this is just about, mushrooms on a pizza or that fuzzy green stuff on the bread you left in the pantry for a week, let me stop you. We are going way deeper.
Way deeper.
We are looking at the structural and conceptual framework of the entire fungal kingdom. This is the foundation. If you don't understand the concepts we break down today, none of the specific diseases, your candidiasis, your aspergillosis, ringworm, none of it will make any sense later on your exams or in the clinic.
That's right. We're building the scaffolding today. So for our listeners following along, we are drawing primarily from Chapter 57 of Murray's Medical Microbiology. That's going to serve as our structural spine.
The roadmap.
The roadmap, yes. And we're also layering in some high-yield mechanisms from Jawetz's Medical Microbiology, specifically Chapter 45, to really flesh out the pharmacology and those clinical patterns where Murray kind of leaves off.
So let's start with the problem. The core issue that defines this entire field of medical mycology, we call it the eukaryotic dilemma.
The eukaryotic dilemma. That's the perfect hook. See, if you're a doctor trying to kill a bacterium, you have a distinct tactical advantage. Bacteria are prokaryotes.
Meaning they're simple?
Simple and evolutionarily very distant from us. They have cellular machinery like 70S ribosomes or these thick peptidoglycan cell walls that human cells simply do not have.
So it's like a sniper taking a shot at a target wearing a bright neon yellow vest in a crowd of people wearing dark blue.
That's exactly it. The target stands out perfectly. You can hit that target, the bacteria, without touching the crowd, which are the human cells. Penicillin attacks peptidoglycan. Humans don't have peptidoglycan. Easy.
But fungi are a different story.
Fungi are eukaryotes, just like us.
Which means if you look at a fungal cell under an electron microscope, it looks disturbingly familiar.
It really does. You see a true nucleus wrapped in a membrane. You see mitochondria generating ATP. You see a rough endoplasmic reticulum. You see a Golgi apparatus packaging proteins. Biologically speaking, fungi are our cousins.
Wow.
Yeah, we are far more closely related to fungi than we are to plants or bacteria. And that shared biology creates the central challenge. How do you kill the invader without harming the host?
That is a high-stakes game, because if you poison the fungus's protein synthesis,
You might poison the human's protein synthesis, exactly. If you attack their DNA replication, you might stop the patient's own cells from dividing.
So the collateral damage is almost guaranteed if you're not careful.
Precisely. This is why antifungal drugs are notoriously more toxic than antibacterial drugs. We are working with a much, much smaller margin of error. We have to find the tiniest microscopic differences between us and them and exploit them ruthlessly.
So our mission for this deep dive is to find those differences. We need to build a mental model of what makes a fungus a fungus. But before we get into the heavy biology, we need to reframe how we actually see them. When I think of fungi in a medical context, I default to germs. I think pathogen. But that's not really their day job, is it?
Not at all. In the grand scheme of things, fungi are the Earth's recyclers. They are environmental degraders. Their primary biological directive is to break down complex organic matter.
Like dead trees.
Dead trees, fallen leaves, insect shells, you name it. They turn all that carbon back into simple nutrients for the soil. They are saprobes.
Saprobes. That's a key term we'll need to anchor on.
Yes. A saprobe is an organism that lives on dead or decaying organic matter. That's what most fungi are doing 99% of the time. The pathogen part, the part where they infect humans, is often an accident. It's an opportunistic shift.
So they're basically, composters that decided to try a new food source.
Ideally, no. Most fungi heavily prefer the soil. But here's where it gets scary. Murray makes a very bold statement in Chapter 57. He says that, essentially, there are no non-pathogenic fungi anymore.
Wait, really? No non-pathogenic fungi? That seems a bit extreme. Surely the little bit of mold on my cheddar cheese isn't going to kill me.
For you, a healthy host with a functional immune system? Probably not. But that statement reflects a massive seismic shift in modern medicine. Over the last few decades, we have created a massive population of immunocompromised hosts.
Ah, the landscape changed.
Exactly. We have patients living with HIV/AIDS. We have patients receiving solid organ transplants who have to be on lifelong immunosuppressants so they don't reject the organ. We have patients on aggressive chemotherapy for cancer. Patients with central venous catheters sitting in their chest.
We've artificially lowered the barrier to entry.
We have. The definition of a pathogen has shifted. It's no longer just about the bug's inherent strength or virulence. It's about the host's weakness. A fungus that is completely harmless to you out in the garden could be absolutely lethal to a bone marrow transplant recipient.
Because there's no immune system to keep the saprobe in check.
Right, so the distinction between a saprobe and a parasite is becoming very, very blurry in the clinical setting.
That is a profound way to look at it. It's all about context. The same organism can be a vital helper in the soil and a rapid killer in the ICU. Okay, let's get into the biological nuts and bolts. Section 1, the fundamental nature of fungi. We established they're eukaryotes. What else defines this kingdom?
The second big rule you need to know is that they are heterotrophs.
Contrast that for us, because plants are autotrophs, right?
Exactly. Plants have chlorophyll, they use sunlight and carbon dioxide to synthesize their own glucose. They make their own food. Fungi are strictly non-photosynthetic. They cannot make their own food.
So they're like us in that way.
They're like animals in that sense, yes.
Yeah.
They have to consume pre-existing organic matter.
But they don't have mouths. They don't chew. How do they eat a tree?
They are what we call absorptive heterotrophs. They secrete these incredibly powerful enzymes, exoenzymes, out into their immediate environment.
Oh, wow.
Yeah, these enzymes digest the food outside the fungal body. They break down those big, tough polymers like cellulose or keratin or protein into tiny, simple molecules. And then the fungus simply absorbs the resulting nutrient soup.
So they essentially digest their food before they eat it. That is incredibly efficient.
It is. It's like a spider wrapping its prey and injecting it with digestive fluids. And depending on their relationship with the source of that food, we categorize them clinically. If they eat dead matter, like we said, they are saprobes. If they live in a neutral or mutually beneficial association with a host, like the fungi in your gut, they are symbionts or commensals. And if they extract resources from a living host to the host's detriment, they are parasites.
So, strictly speaking, when a fungus causes a disease in a human mycosis, it is acting as a parasite.
Correct. But remember, for many of these environmental fungi, that parasitic phase in a human is a dead end. If they kill the host, they eventually have to go back to being a saprobe in the soil. We're often just collateral damage in their blind quest for carbon and nitrogen.
Let's zoom in on the physical structure. Because if I'm a medical student looking at a drug mechanism or a researcher trying to develop a new fungicide, I need to know about the cellular armor. This seems to be where the rubber meets the road for pharmacology.
This is section 2, and honestly, if you zone out for everything else, pay attention here. This is incredibly high-yield territory for exams. Because fungi are eukaryotes, we already established we can't target their ribosomes or their DNA easily without hurting ourselves. So we look at the envelope, we look at the cell wall and the cell membrane.
Let's start with the cell wall. Because humans don't have cell walls at all. Our cells are just squishy lipid membranes.
Right. Bacteria have cell walls made of peptidoglycan. Plants have cell walls made of cellulose. Fungi have a thick, rigid cell wall, but it is chemically totally unique. It is composed primarily of chitin and glucan.
Chitin, I know that word. That's the same material in the exoskeletons of insects and crustaceans, isn't it? Like a crab shell.
It is exactly the same polymer. It's a polymer of N-acetylglucosamine. It forms these long, incredibly tough, rigid fibers that give the fungal cell its shape and protect it from osmotic shock.
Osmotic shock.
Yeah, if you didn't have that rigid wall, the fungal cell would just absorb water from the environment until it swelled up and exploded. The wall contains that pressure.
Okay, so chitin provides the structural steel. What about the glucan?
Glucans are complex polysaccharides, basically long branch chains of glucose. Specifically in medical mycology, we're interested in beta-1-couple-3 glucan and beta-1-couple-6 glucan. These form a sticky matrix that cross-links the chitin fibers.
So if chitin is the steel rebar, glucan is the concrete poured over it.
That is a perfect mental model, rebar and concrete.
And why do we care about beta-glucan specifically? Is it just a structural trivia fact?
No, it is a massive pharmacological target. Teyvat's highlights this beautifully. Beta-1,3 glucan synthesis is the specific target of the newest major class of antifungal drugs, the echinocandins.
The echinocandins, drugs ending in fungin, right? Like caspofungin, mycofungin.
Inidilifungin, exactly.
How do they actually work at the cellular level?
They act as non-competitive inhibitors of the enzyme 1,3-beta-glucan syntase. So essentially, going back to your analogy, they stop the fungus from mixing and pouring the concrete.
Oh, that's brilliant.
Without the concrete, the chitin rebar isn't enough. The wall becomes weak, the cell can't withstand the pressure of its own internal fluids, and it leases, it just pops.
I love that visual, stealing the concrete from the construction site. So echinocandins target the wall. That's target number 1. What about target number 2, the cell membrane?
This is arguably the most important concept in the entire history of antifungal therapy. The cell membrane sits just inside the cell wall. It's a lipid bilayer, very much like ours. It regulates what goes in and out of the cell. Now, in mammalian cells, human cells, the main sterile molecule that sits in that membrane and keeps it stable and fluid is cholesterol.
Right. We all know cholesterol. It keeps our cell membranes from just melting or falling apart.
In fungi, the main sterol doing that exact same job is ergosterol.
Ergosterol versus cholesterol. It sounds like a really minor chemical difference, just a few atoms.
Chemically, they are very similar. They're both these flat, rigid, four-ring structures. But biologically and pharmacologically, that tiny difference is absolutely everything. It is the entire basis for the selectivity of our most powerful drugs.
So how do we exploit it? How do we hit ergosterol without hitting cholesterol?
We have two major weapon systems here. First, we have the polyenes. And the prototype, the grandfather of them all, is amphotericin B.
Amphotericin B, the heavy hitter.
The absolute nuclear option for severe fungal infections. Amphotericin B is this large, ring-shaped amphipathic molecule. One side loves water, one side loves fat. It binds directly to the ergosterol that is already sitting in the fungal membrane.
And then what?
When several of these amphotericin B molecules bind to ergosterol, they aggregate together. They group up and form a physical pore, a literal hole, right through the membrane.
It punches a hole.
Literally punches a hole. It turns the cell membrane into a leaky sieve. Essential intracellular ions like potassium and magnesium leak out. Protons leak in. The cellular machinery shuts down, and the cell dies rapidly. It is fungicidal. It kills them dead.
That sounds incredibly effective. But if you talk to anyone who works in a hospital, they call it amphoterrible. Why does it have such a notoriously bad reputation?
Here is the catch, and it all goes back to the eukaryotic dilemma. Remember I said ergosterol is chemically very similar to human cholesterol.
Uh-oh. I see where this is going.
Amphotericin B binds preferentially to ergosterol, yes, but not exclusively. It has some affinity for human cholesterol. So when you infuse this drug into a patient's vein to kill the fungus in their blood or lungs, it also binds to the patient's own cell membranes, particularly the membranes of the cells in the kidneys.
Nephrotoxicity. It punches holes in the kidney cells.
Exactly. It punches holes in the host, too. It causes severe kidney damage. It also triggers massive cytokine release, which causes high fevers, rigors, chills, what they call the shake-and-bake infusion reaction. It is a highly toxic drug because the target, ergosterol, is just too similar to the host structure, cholesterol.
That perfectly illustrates the dilemma. You're trying to snipe a target that looks almost exactly like the bystanders.
Okay, so polyenes like amphotericin B punch holes by binding directly to ergosterol. What about the other big class, the ones that also involve ergosterol?
The azoles. Fluconazole, itraconazole, voriconazole, posaconazole, these are the daily workhorses of the antifungal world. They don't punch holes. They take a much more subtle approach. They inhibit the synthesis of ergosterol. So they just cut off the supply chain.
Right. They block a specific fungal cytochrome P450 enzyme. It's called 14-alpha-demethylase. This enzyme is required to convert a precursor molecule called lanosterol into ergosterol.
So the fungus can't make new membrane stabilizers.
Exactly. And not only is there a lack of ergosterol, but those toxic precursors like lanosterol build up in the membrane. The membrane becomes stiff, toxic, and unstable, and fungal cell division stops.
Do these kill the fungus dead like amphotericin?
Usually no. The azoles are generally fungistatic against yeasts, meaning they halt growth and let the host immune system clean up the mess. But here's the beauty. They are much less toxic to humans than amphotericin B because they are highly specific to the fungal P450 enzyme.
Though, because they mess with cytochrome P450, don't they cause a ton of drug interactions in the liver?
A massive amount. If a patient is on an azole, you have to check all their other medications because the azole will inhibit human CYP450 enzymes to some degree, changing how other drugs are metabolized. It's a huge clinical pro.
Okay, so to recap the cellular armor, because this is so crucial. We have the chitin and glucan wall, which is like rebar and concrete, targeted by the echinocandins, and we have the ergosterol membrane, which is targeted directly by the hole-punching polyenes or indirectly by the supply chain blocking azoles.
Perfect summary.
Before we move on to what these things actually look like, let's talk about speed, metabolism, and growth. If I'm thinking about bacteria, bacteria are fast. If I have E. coli in a warm broth, it doubles every 20 minutes.
Right. Bacteria sprint. Fungi are tortoises. Their cell doubling time is measured in hours, not minutes. Something like Candida might double every hour or two under perfect conditions, but many environmental molds take much, much longer.
Why does this sluggishness matter clinically?
It matters immensely for the microbiology lab. If a patient comes in and you suspect a bacterial pneumonia, you take a sputum sample, plate it, and you might have a preliminary culture result the very next morning. If you suspect a fungal pneumonia, that culture might take days or even weeks to grow visible colonies.
Weeks? You're waiting weeks with a sick patient.
For some slow-growing endemic organisms like histoplasma, absolutely. It can take up to 6 weeks. This means, as a clinician, you often cannot wait for the lab to definitively prove what it is. You have to treat empirically based on clinical suspicion, imaging, and faster non-culture tests because the fungus is simply too slow to grow on a petri dish.
That's a crucial clinical pearl. Patience is required for the lab, but urgency in treatment is needed for the patient. It's a real paradox.
And while they are growing slowly, they are metabolically very active. As we mentioned, they are heterotrophs. They produce primary metabolites just to live, like ethanol and citric acid.
Wait, citric acid? Like the stuff in sour candy and soda?
Yes, fun fact. Almost all the citric acid used in the global food and beverage industry is not squeezed from lemons. It is produced in massive industrial vats by a mold called Aspergillus niger. They are industrial workhorses.
I will literally never look at a can of Sprite the same way again.
You shouldn't. But they also produce secondary metabolites. These aren't essential for their basic growth, but they give the fungus a distinct survival advantage in the wild. Penicillin is a fungal secondary metabolite. It's a chemical weapon the fungus evolved millions of years ago to kill competing bacteria in the soil.
So antibiotics are just fungal warfare that we learn how to bottle.
Exactly. But they also make mycotoxins, which are not helpful to us. Aflatoxin, produced by Aspergillus flavus, is one of the most potent naturally occurring carcinogens on Earth. It contaminates moldy peanuts and corn. If you ingest it over time, it causes a specific mutation in the p53 tumor suppressor gene, leading to hepatocellular carcinoma liver cancer. So their metabolism is deeply relevant to medicine, industry, and agriculture.
That is fascinating. Okay, let's unpack the morphology. This is section 3. If I take a sample and look through a microscope, what am I actually seeing? I feel like there are two main shapes that people talk about, but then the terminology gets incredibly complicated.
It does get thick with jargon, but let's simplify it first. At the most fundamental level, the two basic morphological forms of fungi are yeasts and moulds.
Yeasts. Okay, I think of baking bread, brewing beer, and, well, yeast infections.
Biologically, a yeast is defined as a unicellular fungus. It is a single, standalone cell. Usually they are spherical or ellipsoidal, kind of oval-shaped. If you streak a yeast onto an agar plate in the lab, the colony that grows looks creamy, opaque, and pasty. Honestly, to the naked eye, a yeast colony looks a lot like a bacterial colony.
And how do they reproduce if they're just single cells?
Usually by budding. We call the resulting cells blastoconidia.
Give me a visual.
Imagine a round balloon. Now imagine a smaller balloon slowly inflating off the side of it. That smaller balloon is the bud. The mother cell's nucleus divides through mitosis. One copy of the DNA goes into the bud, and eventually the cell wall pinches off, and the daughter cell floats away.
Simple enough, mother cell, daughter cell.
But there is a very important clinical variation. Sometimes, under certain stress conditions, that daughter cell doesn't pinch off. It stays attached to the mother. And then it buds, and that new bud stays attached. You get this long, branching chain of elongated cells that looks exactly like a string of sausage links.
The sausage links. I know this has a specific name on board exams.
Pseudohyphae. False hyphae.
Why are they false?
Because unlike true mold hyphae, which we'll talk about in a second, there is a distinct visible constriction, a pinching at the connection point between each cell, just like the twist between two sausage links. True hyphae are like continuous pipes. Pseudohyphae are like chained balloons.
And who famously does this?
Candida albicans. It's a massive diagnostic clue.
Mm.
If a pathologist looks at a tissue sample and sees round yeast cells mixed with these long sausage chain pseudohyphae, they're almost certainly looking at an invasive Candida infection.
Okay, so that's yeast. Single cells, budding, sometimes forming sausage chains. What about molds?
Molds are multicellular. They form these long, thread-like microscopic tubes called hyphae. If yeasts are balloons, molds are garden hoses or bamboo poles.
And they grow differently than yeasts.
Yes, they grow by apical extension, meaning they only grow from the very tip, extending outward like a root. As they grow, they branch and intertwine with other hyphae to form a thick, tangled mat called a mycelium.
And the mycelium is what we actually see.
Right, that's the fuzzy, woolly, or powdery colony you see growing on an agar plate or on that orange you forgot in the back of the fridge. The part digging into the orange to absorb nutrients is the vegetative mycelium. The fuzzy part sticking up into the air is the aerial mycelium, which is making spores to blow away in the wind.
Now, inside those garden hoses, those hyphae, there's an important structural difference that comes up on microbiology exams constantly.
The cross walls, we call them septa.
Unpack this.
Most medically important molds like Aspergillus or the dermatophytes that cause ringworm have hyphae that are regularly divided into distinct cellular compartments by these internal cross walls. We call those septate hyphae.
So that's the bamboo pole analogy. It's one long structure, but it has internal dividers.
Exactly. The septa have tiny pores in them to let cytoplasm and nutrients flow down the tube, but there is a structural partition. But there is one major group of fungi, the mucorales or mucormycetes, that essentially lacks these walls.
The mucor group.
Right. Their hyphae are just these long, continuous hollow tubes filled with cytoplasm and hundreds of nuclei floating around. We call that choanocytic or posiseptate, meaning very few septa.
Okay, so if septate hyphae are bamboo poles, choanocytic hyphae are just plain garden hoses. Why does that matter structurally or clinically?
Think about the plumbing. If you puncture a bamboo pole, a septate hypha, the damage is contained to just that one section. The cross walls seal it off. The rest of the hypha survives. If you puncture a garden hose, a choanocytic hypha, the whole thing loses pressure, leaks out its cytoplasm, and collapses.
So the mucorales are physically fragile.
In the lab, yes. If you are rough with a tissue sample, if you grind it up too hard before plating it, you might destroy all the choanocytic hyphae, and the culture will come back negative, even though the patient is infected. But in the body, this open, wall-less structure allows for incredibly rapid transport of nutrients from one end of the fungus to the other.
Because there are no toll booths? No walls to slow the flow down?
Exactly. Mucorales grow incredibly fast. In the lab, we call them lid lifters because the fuzzy mycelium grows so rapidly and so high, it can literally push the lid off the petri dish in a couple of days.
That is a terrifying and great mental image. Bamboo versus garden hose.
Okay, so we have yeasts and we have molds, but then we have the rule breakers, the shapeshifters.
The dimorphic fungi. This is Section 3's grand finale, and it's essential. The Jekyll and Hyde mechanism.
These are fungi that can switch between being a mold and being a yeast. What triggers the switch? Is it food?
It's temperature. That is the golden rule you must remember. Thermal dimorphism.
We have a medical school rhyme for this, don't we? We do. Mold in the cold, yeast in the beast.
I love that. Unpack how that actually works in nature.
In the environment, out in the soil, in a bat cave, on rotting wood, the temperature is ambient, usually around 25 degrees Celsius. At this cooler temperature, the fungus expresses a specific set of genes to grow as a mold, makes hyphae, it forms a mycelium, and it produces spores.
And this environmental mold phase is actually the infectious phase, right?
Yes, because the mold produces those microscopic spores that can float in the air. You walk by, you kick up some dirt, and you inhale them.
And then what happens?
The spores land deep in your lungs. Suddenly, the temperature is 37 degrees Celsius. That sudden heat shock acts as a trigger. It flips a genetic switch in the fungus. The fungus stops being a mold, it drops its hyphae, and it converts into a yeast. Or in the case of coccidioids, a yeast-like structure called a spherule.
Well, why bother changing shape? Why not just stay a mold in the lung and grow hyphae?
It's a highly evolved survival and virulence strategy. Molds are big, long, and fuzzy. They are very easy for the immune system, specifically macrophages and neutrophils, to recognize, trap, and destroy. Yeasts are small, single cells. They can hide inside macrophages. They can evade the immune system and hitch a ride in the bloodstream to disseminate to the brain or the bones.
So it's a tactical adaptation. It's camouflage.
Precisely. This thermal dimorphism is the defining hallmark of what we call the endemic mycoses. Pathogens like histoplasma, blastomyces, and coccidioids.
So if I'm a microbiologist looking at a culture of histoplasma growing on a bench at room temperature, it looks like a fuzzy mold. But if I look at a biopsy slide from that same patient's lung, I'm going to see little round yeast cells.
Exactly. It's the exact same organism, just wearing a different coat. And often, to definitively prove the diagnosis in the lab, we will take that fuzzy mold from the 25-degree incubator and move it to a 37-degree incubator to see if it physically converts into a yeast. If it converts, we know we have a dimorphic pathogen.
Incredible. Let's move to section 4. Reproduction. Fungal sex. Or, I guess, lack thereof.
It's both. Fungi are incredibly versatile. They can reproduce sexually, which involves meiosis and the fusion of compatible mating types to mix DNA, or they can reproduce asexually, which is just mitosis cloning themselves over and over.
And this duality leads to one of the absolute most confusing, hair-pulling aspects of medical mycology, the naming system. Why on earth do fungi have multiple names?
Oh, the anamorph versus teleomorph disaster. It's a historical accident, really. See, before we had cheap, rapid DNA sequencing, mycologists classified fungi solely by how they looked under the microscope. Morphology was king.
Okay. That makes sense for the 1950s.
Right. So a doctor would isolate a fungus from a sick patient. In the lab, it was reproducing asexually, just cloning itself via spores. The mycologist would describe it, draw it, and name it. Let's say they named it Histoplasma capsulatum. This asexual, clinically isolated form is called the anamorph.
The anamorph, got it. This is the clone.
Then, maybe decades later, an environmental biologist will be studying fungi out in the soil under very specific, harsh conditions, and suddenly they'd find a fungus that was having sex. It was producing sexual fruiting bodies and sexual spores. Because these sexual structures look completely, radically different from the asexual spores, the biologist assumed it was a brand new, undiscovered species. So they gave it a completely different name, like Agelomyces capsulatum. This sexual form is the teleomorph.
Let me get this straight. Histoplasma and Agelomyces are the exact same organism.
Yes, they are literally the same biological entity. One is just the name for the asexual clone phase, the anamorph, and the other is the name for the sexual phase, the teleomorph.
That is maddening for students trying to memorize this stuff.
It was a nightmare. But recently, we had a taxonomic revolution, the one fungus, one name rule. Molecular taxonomy, sequencing the ribosomal RNA, proved they are the exact same organism. So the international governing bodies finally stepped in and said, enough. The rule now is, we generally use the name of the form most commonly encountered in clinical practice and culture.
Which is almost always.
The anamorph, the asexual form. Because fungi rarely bother having sex in the cozy, nutrient-rich environment of a human body or an agar plate. So clinically, we stick with the name histoplasma. You can largely ignore the teleomorph names like Agelomyces unless you are a hardcore taxonomist.
Thank goodness for DNA sequencing. Now, let's talk about the spores themselves. Because looking at the asexual spores under the microscope is exactly how we identify these molds in the clinical lab.
Right. We focus on the asexual spores. And structurally, we break them down into two broad visual categories, sporangiospores and conidia.
Give me a solid visual anchor for sporangiospores.
Think of a lollipop.
A lollipop.
You have a long, straight stalk, which is called the sporangiophore. And at the very top of the stalk is a round, enclosed sac called the sporangium. The actual spores are packed tightly inside that sac.
So it's like candy enclosed inside a plastic wrapper.
Exactly. The spores develop inside, and when they are mature, the sac bursts open, the wrapper breaks, and all the spores spill out into the environment.
And clinically, who has these lollipop spores?
The mucorales, those fast-growing coenocytic fungi we talked about earlier, genera like Rhizopus and Eucor. If you look down the microscope and see lollipops, you should immediately think mucorales.
Got it. Lollipops equal mucor. And what about the other type, conidia?
Conidia comes from the ancient Greek word for dust. These spores are what we call naked. They are not enclosed in a sac. They grow completely exposed to the air, pinching off directly from specialized stalks called conidiophores.
Give me a visual for the naked spores.
Think of a dandelion that has gone to seed. You have a stalk and then a fluffy puffball of seeds just sitting there, completely exposed, ready to be blown away by the slightest breeze. Or think of a paintbrush with bristles.
Naked spores. Dandelions and paintbrushes.
This represents the vast majority of medical molds. This is your aspergillus. In fact, aspergillus looks so much like a holy water sprinkler used in Catric Mass and Aspergillum that that's how it got its name. Conidia is also the form used by penicillium, which looks like a bony skeleton hand or a paintbrush, Right. and the dermatophytes that cause ringworm.
Okay, so lollipops are sporangiospores for mucor. And dandelions, Arcanadia for aspergillus, and almost everyone else, that really helps visualize it.
It's a very simple microscopic distinction, but it instantly separates two massive groups of fungi when you're trying to make a diagnosis.
Which leads us perfectly into Section 5, taxonomy. We have 4 major phyla to worry about, according to Murray. We don't need to be evolutionary botanists here, but we need to know the clinical buckets.
Exactly. Taxonomists group them by how they reproduce sexually, even if we clinicians only care about the asexual stuff. Let's start with the one we just hinted at, the Glomeromycota.
That's a mouthful, also known historically as the Mucormycetes.
Right. These are often considered the more primitive molds.
What are the high-yield keywords for this group?
Broad, ribbon-like, aseptate, or coenocytic.
Ah, the garden hoses.
Yes, the garden hoses. And their reproduction is via those lollipop sporangiospores. The key genera you'll see on exams and in the hospital are Rhizopus, Mucor, and Lichthemia. Oh, and functionally, some of them have these little root-like structures called rhizoids that they use to anchor into the agar. The presence and location of those roots help the lab tell Rhizopus apart from Mucor.
And clinically, why do we fear this phylum?
These are the agents of mucormycosis. This is a terrifying, rapidly fatal medical emergency. It is an angioinvasive infection, meaning the fungus literally eats its way into and through the walls of your blood vessels, causing massive tissue necrosis and black dead tissue.
Who gets this? It's not healthy people.
Rarely. It is classically seen in poorly controlled diabetics who are in diabetic ketoacidosis or DKA, or in severely immunocompromised patients like leukemia patients.
Why diabetic specifically?
Rhizopus absolutely loves high glucose and highly acidic environments, which perfectly describes the blood in DKA. Furthermore, in DKA, the acidity causes iron to unbind from transport proteins, leaving free iron floating in the blood. Free iron acts like a super fertilizer for Rhizopus. It grows explosively, it often starts in the sinuses, and eats right through the bone into the brain.
That is horrific. Broad ribbons equal mucor equal diabetic emergency. Okay, let's move to the next group. Basidiomycota.
The Basidiomycetes. In the forest, this group includes the classic toadstool mushrooms, puffballs, and those hard-shelf fungi growing on tree trunks. But medically, we don't really worry about mushrooms unless you eat a poisonous one. Medically, we care about the yeasts that happen to belong to this group. The absolute superstar here is Cryptococcus.
Cryptococcus neoformans.
Correct. Now, even though Cryptococcus grows as a single-celled budding yeast in the human body, its DNA and its sexual cycle prove that taxonomically, it's a Basidiomycete mushroom cousin.
Is there a visual giveaway for this group if we happen to look at their filamentous environmental forms?
There is. It's a unique microscopic structure called a clamp connection. It's a little looping tube that forms between cells during division to let nuclei bypass each other. It literally looks like a tiny little handle or a plumbing bypass loop on the side of the hyphae. If you see clamp connections, you are definitively looking at a Basidiomycete.
But for Cryptococcus the yeast, in a patient, we mainly just look for its armor, right?
Right. It has a massive, thick polysaccharide capsule that repels the immune system. Jowett highlights that it's the only fully encapsulated fungus of major human significance. You diagnose it using an India ink stain of spinal fluid, where the capsule looks like a clear halo against a black background. But knowing it's a Basidiomycete helps you understand its environmental life cycle, where it produces sexual basidiospores.
Okay, third group. And this is the big one, Ascomycota.
The Ascomycetes. This phylum is the big tent. It accounts for about 80% of all medical fungi. Honestly, if you are guessing on a multiple-choice exam and don't know the phylum, guess Ascomycete. You'll usually be right.
Who's in this club?
Almost everyone else we care about. Candida, Aspergillus, all the dimorphics like histoplasma and blastomyces, the dermatophytes.
So it contains yeasts, moulds, and the shape-shifting dimorphics.
All of them. They are defined botanically by producing sexual spores inside a tiny sac called an ascus. But again, clinically, we almost exclusively see their asexual conidia, the naked dandelion spores.
And there was a very special, historically confusing member of this club that had a bit of an identity crisis, didn't it?
Oh, Pneumocystis girovecii.
The organism formerly known as a protozoan.
This is a classic medical history and exam story. For decades, the entire medical community thought Pneumocystis was a parasite. They thought it was a protozoan, related to things like trypanosomes or amoebas.
Why did they think that? Didn't it look like a fungus?
It didn't act like one. It absolutely refuses to grow on standard fungal agar in the lab. Under the microscope, it has a trophic form and a cyst form, which is classic parasite terminology. And most importantly, It responds clinically to antiprotozoal antibiotics, like Bactrim trimethoprim sulfamethoxazole. It does not respond at all to our big antifungal drugs like amphotericin B.
So if it looks like a parasite and dies like a parasite, why do we call it a fungus now?
DNA changed everything. When we finally managed to extract and sequence its ribosomal RNA, it didn't align with parasites at all. It landed smack in the middle of the Ascomycota phylogenetic tree right next to the yeasts.
You can't argue with the genetic code, but why doesn't amphotericin B work on it then?
Because it's a highly adapted fungus that happens to lack ergosterol in its cell membrane. It uses cholesterol it steals from the host. Since ampho-B targets ergosterol, the drug is useless.
Wow. So pneumocystis is a fungus, specifically an ascomycete. That is a crucial gotcha fact for boards.
Absolutely.
Let's hit the fourth and final group, the microspora.
The microsporidia. These are easily the weirdest of the bunch. Like pneumocystis, they used to be classified as protozoan parasites for a long time.
What makes them so weird?
They're obligate intracellular parasites. They absolutely cannot live or multiply outside of a host cell. They are tiny unicellular organisms, and they form these incredibly tough environmental spores.
How do they actually infect a human cell?
This is where it gets crazy. They have a completely unique mechanism. Inside the spore is a tightly coiled structure called a polar tubule. When the spore is swallowed and reaches the gut, it triggers. It shoots this polar tubule out like a microscopic harpoon.
A harpoon? That sounds like science fiction.
It really does. The harpoon pierces the host cell's membrane, acts like a hypodermic needle, and the fungus injects its entire nucleus and cytoplasm directly into the host cell.
That is aggressive. But again, DNA proved they belong in kingdom fungi.
DNA and the discovery of our old friend chitin in their protected spore wall, they are now considered highly specialized, degenerate fungi, closely related to an ancient group called the cutrids.
And clinically, who gets infected by harpoon-shooting fungi?
They are prominent opportunistic pathogens in patients with advanced AIDS. They typically cause chronic, severe wasting diarrhea. You'll see general names like enterocytosome and encephalidosome.
It's amazing. Even the things that don't look or act anything like fungi are turning out to be fungi. The kingdom is just expanding.
It's a very big tent once you look at the genetics.
Let's transition to section 6. We have the biology down, we have the structural taxonomy, and now let's organize this the way a doctor actually thinks. If a patient walks into the clinic, we don't classify their infection by phylum, we classify it by where is the infection.
Right. Clinical classification is strictly anatomical. We start from the absolute outside of the body and we work our way in.
Layer 1, superficial mycoses.
This is the outermost boundary, the absolute dead keratinized top layer of skin or the outside of a hair shaft. These infections are entirely cosmetic. They don't hurt, they don't destroy living tissue, and importantly, they do not trigger a cellular immune response. Your immune system basically ignores them.
Give me a classic example.
Pityriasis versicolor, also known as Dynia versicolor. It's caused by a lipophilic yeast called Malassezia. It causes these patchy, scaly areas of hypo or hyperpigmented skin, often on the chest or the back of young adults. The fungus is just happily feeding on skin oils, specifically sebum.
Oh, this is the one that looks like spaghetti and meatballs under the microscope.
Yes, the mix of short hyphae and round yeast cells. It's annoying, it messes with your tan, but it is not dangerous at all.
Okay, layer 2, cutaneous mycoses.
Now we are digging a tiny bit deeper. We're still in the keratized layers, the stratum corneum of the skin, the full thickness of the nails, the hair follicles. These are the domain of the dermatophytes.
Cotineous.
Exactly. Tinea pedis, which is athlete's foot. Tinea corporis, which is ringworm on the body. Tinea curis, jock itch. These fungi, primarily the genera Trichophyton, Microsporum, and Epidermophyton, have specialized enzymes called keratinases.
They literally eat keratin.
They digest the tough structural protein that makes up your skin, hair, and nails. And because they are actively breaking down tissue and releasing fungal metabolites, the host immune system notices, it reacts. That's why a ringworm lesion is red, raised, itchy, and scaly at the border. It's an active inflammatory response trying to push the fungus out.
Okay, so the rule of thumb, superficial equals no inflammation. Cutaneous equals tissue destruction, inflammation, and itching.
Correct.
Layer 3, subcutaneous mycoses.
Deeper still.
Uh-huh.
Now we are into the dermis, the subcutaneous fat, the fascia, and the muscle. These infections don't just happen by brushing up against a fungus. The skin is too good a barrier. You need a portal of entry.
To mama.
Exactly. Traumatic inoculation. A splinter, a thorn prick, a deep cut from a contaminated tool. The environmental fungus is forcefully implanted deep into the sterile tissue.
What's the classic textbook example for this?
Sporotrichosis, also known as rose gardener's disease. You are out pruning roses, you get pricked by a thorn that is contaminated with a dimorphic fungus, Sporothrix schenki. It implants deep in the finger or hand. It causes a painless nodule that ulcerates, and then the fungus slowly spreads up the lymphatic channels of the arm, causing a chain of new draining ulcers as it goes.
That sounds awful, but it generally stays localized to that one arm, right?
Usually, yes. The core body temperature and the immune system usually keep it from going fully systemic to the internal organs, unless the host is highly immunocompromised.
Which brings us to the scary stuff, layer 4. The endemic mycoses.
Also called the systemic mycoses. These are the true pathogens.
Define true pathogen again in this context, just to be crystal clear.
It means they have the inherent virulence to infect and cause disease in a completely healthy, immunologically normal host. You do not need to have AIDS or a transplant to get sick from these. If you breathe in enough coccidioid spores during a dust storm in Arizona, you are going to get pneumonia, even if you are a completely healthy Olympic athlete.
And these are the ones deeply tied to geography, right?
Yes, their environmental mold forms require very specific soil conditions, humidity, and temperatures to thrive. Histoplasma is endemic to the Ohio and Mississippi River valleys, heavily associated with nitrogen-rich bird and bat guano. Blastomyces is found in the southeast and Midwest, often associated with rotting wood and riverbanks. Coccidioids loves the arid alkaline desert soil of the American Southwest.
And the mechanism of infection for all of these is that thermal dimorphism we talked about earlier.
Exactly. You inhale the mold spores from the environment, they hit the 37-degree heat of your lungs, and they convert into the parasitic yeast or seral phase. And from the lung, they can disseminate via macrophages to the bones, the skin, or the meninges.
Finally, we arrive at layer 5, the opportunistic mycoses.
This is by far the largest, most clinically relevant, and fastest-growing category in modern hospitals. These are fungi that are either commensals, meaning they already live inside us peacefully, or they're ubiquitous environmental molds that we interact with every single day. In a healthy person, they usually do absolutely nothing until the host defenses drop. Broad-spectrum antibiotics wipe out the protective bacterial flora. Corticosteroids paralyze the macrophages. Chemotherapy destroys the neutrophils. An HIV infection wipes out the T cells. Or a surgeon puts a piece of plastic, a central line, or a joint replacement right into the body.
Who are the big players you see in the ICU?
Candida is number one globally. It lives in your gut and mouth naturally. But if you suppress the immune system or wipe out the bacterial competition, Candida overgrows, crosses the mucosal barrier, and invades the bloodstream. We call that candidemia, and it has a shockingly high mortality rate.
And Aspergillus.
Aspergillus is an environmental mold. It's on decaying leaves, it's in dust. You are almost certainly breathing in Aspergillus spores right now as we speak.
Comforting.
But your alveolar macrophages are just quietly eating them up. No problem. However, in a profoundly neutropenic patient, someone whose bone marrow has been wiped out by leukemia treatment, there are no white blood cells to stop it. Those spores germinate in the lung, form hyphae, and literally invade the pulmonary blood vessels, causing massive hemorrhage.
And we already mentioned Cryptococcus, right?
Yes, Cryptococcus neoformans. It's environmental, strongly associated with pigeon droppings. You inhale it. In a healthy person, it causes a mild self-limiting lung infection you might not even notice. But in a patient with advanced AIDS, where the CD4 T cell count has crashed, it disseminates rapidly to the brain and causes a lethal meningoencephalitis.
It really is a sobering list. It perfectly reinforces that concept we discussed at the very beginning. The host's immune status completely defines the pathogen.
Absolutely. The line between a harmless environmental recycler and a lethal invasive pathogen is entirely drawn by the strength of the human immune system.
So we've covered the fundamental eukaryotic nature, the cellular armor, the morphology, reproduction, taxonomy, and the clinical groupings. That is a massive, incredibly dense framework.
It is a lot to take in. But if you hold tightly onto those key mental anchors, the ergosterol target for drugs, the dimorphism switch for systemic disease, the specific wall composition you can hang on occasions, you can hang all the specific diseases and drugs on that framework later. It makes the rest of mycology so much easier to digest.
Before we wrap up with our rapid recall, I want to touch on something Javits mentions very briefly towards the end of the chapter. It's a bit of a provocative thought for the future of medicine, the idea of microevolution and rapid adaptation.
Well, this is fascinating, and frankly, it keeps infectious disease doctors up at night. We tend to think of fungi as these slow, static, unchanging organisms, but they possess massive, highly plastic genomes. They're evolving rapidly under pressure.
Specifically regarding drug resistance, right?
Yes. We are seeing the terrifying emergence of true superfungi. The global poster child for this right now is Candida auris.
I've seen the CDC warnings and the headlines about C. auris. What makes it so bad?
It practically appeared out of nowhere about 15 years ago, simultaneously on 3 different continents. It is highly multi-drug resistant. Some strains are pan-resistant, meaning they're resistant to all 3 major classes of antifungals, the azoles, the polyenes, and the echinocandins.
That means we have literally no approved drugs to treat it.
None. And it behaves entirely differently from normal Candida. Most Candida infections, like we said, are endogenous. They come from the patient's own gut. Candida auris acts more like a hospital superbug, like MRSA. It colonizes the patient's skin, it sheds onto hospital bed rails and blood pressure cuffs, and it survives incredibly harsh disinfectants. It spreads room to room, person to person.
Why is this happening now? Where did it come from?
There are a few compelling theories. One is global warming, the idea that as ambient environmental temperatures rise, environmental fungi are slowly adapting to survive at temperatures closer to 37 degrees Celsius, breaking the thermal barrier that used to protect us.
Wow, so they're pre-adapting to human body heat.
Exactly. Another major theory is agricultural. We use massive, staggering amounts of azole fungicides on our crops worldwide to prevent rot. By soaking the soil in these chemicals, we are putting immense evolutionary pressure on the environmental saprobes. We are effectively training them to survive our best medical drugs before they ever even enter a human host.
We are forcing the harmless saprobes to become highly resistant superparasites.
It is a very distinct possibility. The boundary between the natural environment and the sterile clinic is completely dissolving.
That is a heavy, sobering thought to end on, but an incredibly important one. Fungi aren't just passive background noise. They are dynamic, they are adapting, and they are incredibly persistent.
And above all, they are eukaryotes. Dealing with them is dealing with a distorted, resilient reflection of our own biology. That is the true challenge of medical mycology.
Okay, let's shake off the existential dread for a minute and lock in this knowledge. It is time for the rapid recall.
Let's do it.
I'm going to ask 5 single best answer style questions based closely on what we just discussed. I will pause after reading the prompt, giving you a moment to think or shout the answer at your dashboard, and then our expert will reveal the answer and the mental anchor. Question 1. A patient with a severe systemic infection is treated with an intravenous antifungal drug that selectively binds to sterols in the cell membrane, aggregating to form pores and causing rapid leakage of intracellular ions. Which specific sterol is the primary target of this drug?
The answer is ergosterol. Context here. This is the precise mechanism of the polyene class, specifically amphotericin B. Remember the core rule. Humans have cholesterol to stabilize our membranes. Fungi have ergosterol. That single molecular difference is the basis for the drug's selectivity. You can review this in Murray Chapter 57 under Fungal Taxonomy, Structure, and Replication.
Question 2. A fungal pathogen isolated from a cutaneous skin infection is observed under the light microscope. The pathologist notes multicellular, long, filamentous structures that contain distinct, regular cross walls dividing the tubes into compartments. How are these microscopic structures best described?
The answer is septate hyphae. Context. Septate literally means having a septum, a cross wall, or partition. Think of the bamboo pole. If they lacked those regular walls and were just hollow tubes, they would be described as coenocytic or aseptate, which is characteristic of the fast-growing Eukaryotic. Review this in Murray Chapter 57 under Morphology.
Question 3. A dimorphic fungus is cultured from the sputum of a patient who recently traveled to the Ohio River Valley. If the laboratory maintains this culture in an incubator set at 25 degrees Celsius, which is room temperature, what morphological form will predominantly be observed on the agar plate?
The answer is the mould form.
Context.
You have to remember the rhyme. Mould in the cold, which is 25 degrees, and yeast in the beast, which is 37 degrees. In the ambient environment, or the cold incubator, these dimorphic fungi grow as fuzzy filamentous molds to produce infectious spores. Review this in Murray Chapter 57 under the endemic mycoses section.
Question 4. A patient in diabetic ketoacidosis presents with a rapidly progressing necrotic sinus infection. The biopsy reveals broad, ribbon-like, predominantly aseptate hyphae. Which major fungal phylum is characterized by these structures and reproduces asexually via sporangiospores contained inside a sac?
The answer is the glomeromycota, also known historically as the mucormycetes. Context. The clinical vignette screams mucormycosis. The visual keywords to anchor on are broad, ribbon-like, aseptate, and those lollipop-like sporangiosspores. This points directly to the order Mucorales, which includes Rhizopus and Nucor. Review this in Murray Chapter 57 under the taxonomy of Glomeromycota.
Question 5. An organism causing severe progressive pneumonia in an AIDS patient was historically classified as a protozoan parasite for decades, but it is now definitively classified as an Ascomycete fungus based on ribosomal DNA sequencing. What is the name of this specific organism?
The answer is Pneumocystis girovecii. Context. This is the classic reclassification board question. Clinically, it behaves like a parasite, and it uniquely responds to antiprotozoal drugs like Bactrim rather than traditional antifungals because it lacks ergosterol. But genetically, it is absolutely a fungus. Review this tricky exception in Murray Chapter 57 under Ascomycota.
How did you do? If you miss any of those, take a few minutes to go back and review Murray Chapter 57, specifically paying attention to the sections on structure, dimorphism, and taxonomy.
And honestly, it is highly recommended to take a quick look at the microscopic images in Yeasts Chapter 45 to really cement those visual distinctions between the lollipop sporangia and the dandelion conidia. Seeing it makes it stick.
That is it for this deep dive into the foundational framework of the fungal kingdom. We have built the scaffolding. In future deep dives, we are going to start filling in that structure with the specific terrifying diseases these organisms cause. Until then, keep recycling. But maybe leave the decomposition to the professionals in the soil.
Stay curious, everyone.
See you next time.